Well, good evening. Welcome to Sierra Nevada Forums. Tonight the topic is medical research and breakthroughs at where? University of Nevada, Reno, School of Medicine. So we're very happy to have them here tonight. And I'm going to give you a few house cleaning rules first. The exit right here, along with the door that you came in on. The restroom, if you go up there and around the corner this way, that's where you will find those. And let's see, I think what else I need to tell you guys. These forums are free, and I want to thank you guys who donated in the donation jar, because that's what keeps us afloat for everything that we do, including Ken over here. He is volunteering his services to tape all of this for us. So, and usually you've got them going back and available on the computer.
7 months.
For 7 months now. And it only takes you like a couple days to get them up, right? One day. Hey, good. All right, I would like to go ahead with our introductions tonight, and it begins with Danielle Eaton. And I'm going to read this, and you're going to have to really concentrate hard on this. In her role as Senior Director of Research Operations and Administration, Danielle Eaton works closely with UNR medical students. She was telling me a little bit about that. Residents and faculty to provide a range of support services, such as assuring compliance with human research protections and funding agencies. She is the administrative director of new clinical research center on campus, which seeks to provide a one-stop shop and support infrastructure for getting clinical research projects off of the ground and to the patients. Danielle also serves as a strategic business partner for the Senior Associate Dean of Research, the Associate Dean for Clinical and Thank you. Research, the Basic Science Department chairs, and the Senior Associate Dean for Administrative Finance. She's doing all of this on her science degree, her Bachelor of Science degree in Biology from UNR. And she puts on here, and I find this kind of hilarious, you're working on earning your Master's in Business Administration, and my advice to you is you just tell UNR, you're skipping right over that, and you want 3 PhDs.
I need the letters out.
3 PhDs, you can be the flagship for them. You have an amazing resume, I'm just having a hard time. Yeah, we'll support her, right?
Absolutely, thank you. We'll clap whenever needed.
Come on up, Danielle, show them your stuff.
Thank you. That was an awesome introduction. Can I have you?
All right, I'll call you. Good evening, and thank you for having us. This has been kind of in the works for a little while. Judy reached out to me a year ago to try to put something like this together, and it wasn't until now that we were able to kind of figure out what kind of researchers we could get here and when it worked and the schedule and stuff like that. So we're really appreciative to be here tonight. With that said, my job basically boils down to supporting scientists in doing their sciencey things and taking away the administrative burden that often comes with being brilliant. This set of researchers is just a taste of who we have at the University of Nevada, Reno School of Medicine. We've been celebrating 50 years of great basic science research and outstanding teaching to our medical students. And now we're reaching into the clinical research and translational space with some of our current endeavors and future recruitments. So it's really an exciting time to be at the School of Medicine. I put together this panel, so it's my fault. So we're going to start off with 2 of my favorite kind of researchers who do research in a rare disease, in a rare form of muscular dystrophy. So I will let Dr. Ryan Wuebbles, who is a research assistant professor at the School of Medicine, kind of tell you a little bit about what he does, and then we will talk about his mentor, Dr. Dean Birkin. So Ryan, please come up.
Come up there? Or?
Yeah.
That's where the clicker is. Oh, I'll bring you the clicker. Take mine.
Does this work?
Yeah.
It's always nice when I don't have to walk. Okay, so I'm going to tell you a little bit about my story. Try to keep it high level, less sciencey, and just keep it broad. So I'm going to tell you about how I've been developing therapeutics to treat patients living with muscular dystrophies. I have muscular dystrophy myself, and so it's kind of a personal journey, although I'm going to stick mostly to the professional side of the journey. So this work's been done at UNR, and we've also...
Pointed at me.
Yeah, no, I was sorry. And then we've also formed a company that you'll hear a little bit about called Transgenic in order to try to transition the therapeutics that we've developed from the bench top into the clinic. So brief bio, I was diagnosed with a rare form of muscular dystrophy called FSHD, stands for facioscapulohumeral muscular dystrophy, when I was 20. It's 2000, so you can do the math. So immediately, I was an undergrad at the time, and immediately upon the diagnosis, I read about it, I looked in papers, I looked at what my life and health trajectory was going to look like, and I immediately switched out of electrical engineering and into biology because I said, you know, unless someone really starts pushing therapeutic cures for this disease, it's just never going to happen, and I want to be part of that. So in graduate school, I actually stayed at University of Illinois, and I approached a new professor who had just arrived from NIH, and his name was Dr. Peter Jones. And I said, you know, you study something that I think is involved in FSHD called epigenetics. It involves basically the shutting off of specific genes in the specific context of specific cells. So it's how different cells in your body look and act different. Because every cell in your body has exactly the same genome, the same set of DNA. So how do you control your eye from your foot? Those need to be controlled through epigenetics. So I said, hey, I want to work for you, and I want to do a project based on FSHD. He said, well, you know, come on in and let me introduce you to other epigenetic diseases, but eventually I'll let you work on FSHD. So 4 years in to my study with Dr. Peter Jones, I was getting weaker, but I started a project on FSHD, and since then he has actually taken the kind of side project I started and developed it into, he now runs his full lab on it, and he's one of the leading researchers in FSHD research in the entire world. So pretty proud that I actually helped create at least his lab pushing therapeutic development for FSHD forward. So as a postdoc, I actually transitioned, and I still work with Dr. Dean Birkin. And part of the reason I transitioned here to work at UNR was I wanted to learn how to develop drugs, and that's what Dr. Burkin was doing. We work in his lab on Duchenne Muscular dystrophy, which is a slightly more severe form of muscular dystrophy. I'm going to let him talk more about it. And along with Laminin 2 congenital muscular dystrophy. But I'm getting back into FSHD research now because Dr. Peter Jones has actually now moved to UNR, and we're now forming kind of a muscular dystrophy research center. That's kind of the overarching goal for UNR, I think. Okay, so we then developed a bunch of therapeutics that will help small molecules and recombinant proteins that will actually help treat Duchenne muscular dystrophy, and we've been trying them in other muscular dystrophies as well. And we created a company in order to transition, as I already said, called Stricogen. And if you want to read a little bit more about my story, there was actually an article written in the Synapse that's produced by UNR, Synapse magazine, in 2018. I think it's a spring and summer. So FSHD, it is widely considered to be the most complicated human genetic disease around, and that's because it actually involves the loss of repression of a retrotransposomal element that actually jumped in to primates and is repeated multiple times at the 4q35 locus. It's supposed to be shut down because it encodes a highly toxic protein that, when it's expressed, basically kills every cell that it gets expressed in. And so that's where the epigenetics comes in. So as repeats contract, the ability of our cells to actually keep those elements repressed, we lose those abilities. And so under 10 repeats at this locus of D4Z4 repeat elements, you start getting small amounts of this highly toxic protein called DUX4 expressed. And that leads to the loss of muscle over time. It can occur relatively quickly if you have only a couple of repeat elements, and you see some children that actually will be immobile by the age of 10. But in most cases, I'm pretty standard moderate case, you just gradually lose the ability to do specific functions in your life. I lost my ability to lift my arm over the head when I was somewhere in the 20s. I stopped being able to run as fast when I was 17, 18. Sports, even though I was highly athletic when I was a kid, became harder and harder. And so it's just a downhill trajectory, and you just kind of deal with it. You'll lose an ability, and then you'll plateau for 3 or 4 years. And that's pretty much how the trajectory occurs. Primary affected muscles, although it's highly variable, and even siblings with the same mutation will have an unaffected sibling and a severely affected sibling. So it's highly variable and very difficult to fully understand why the penetrance is what it is. But typically in the biceps, the face, as the name describes, the humerus muscles, the abdominal, and the scapular muscles. My legs, and this is actually a finding that researchers have been describing more and more, is that the legs are actually affected more than previously believed. So here's a little bit about the genetics. A healthy individual has silencing proteins at the 4q35 locus. These are meant to be D4Z4 repeats. So its protein is not expressed. When you get a reduction or a contraction in the repeat array below a threshold of 10 copies, you begin to express small amounts of this DUX4 mRNA, which becomes translated into the DUX4 protein, which kills your cell. There is another form that involves a mutation in a protein that's meant to silence this region, and that's how we know for sure it is actually DUX4 that's causing these problems, because these patients, although it's even more rare than the 1 in 10,000 that FSHD1 is, these patients also have the same muscular dystrophy phenotype. So now I'm going to transition away and back into what we're currently doing in the Burkin lab, and I'm going to go forward because Dean's going to describe these in more depth. But basically, this is the muscle membrane, and these are all proteins that, if you have a mutation in them, will lead to muscular dystrophy of some type. And there's actually over 100 different types of muscular dystrophies now, based on the different types of proteins that are mutated. So one important thing, our hypothesis, and what we work on is we work on enhancing the levels of alpha-7 integrin as a modifier for Duchenne muscular dystrophy progression. And basically, dystrophin is right here. This is what's mutated in the children that have Duchenne muscular dystrophy. And so you lose the ability to connect your actin cytoskeleton up to the extracellular matrix and laminin. Integrin actually does exactly the same role, it's just not quite as strong a protein. But if you overexpress it, Dean always describes it like you're trying to replace Superglue with Elmer's, but Elmer's is better than nothing. So we've done a lot of work, and this goes all the way back to Dr. Birkin's work at Illinois, where I actually originally met him in 2001, in order to prove that if you overexpress alpha-7, it indeed does ameliorate disease progression in these patients. So how do we safely and effectively increase alpha-7 in muscular dystrophy patients? Well, the obvious way, and the way most drug companies are actually looking to do this, is through small molecules. Now, with gene therapy, you're going to want to directly replace the mutated protein. So in this case, dystrophin. But when you use gene therapy to actually replace dystrophin, you're going to create a different kind of muscular dystrophy. And so by increasing the levels of alpha-7, we're actually going to be able to further help those patients. So we developed a drug-based, cell-based assay in order to screen 430,000 compounds, and basically we found a bunch of hits. These are all small molecules that, when we used in tissue culture, will lead to higher levels of alpha-7. We then defined what those small molecules look like in terms of a dose response and the level of activity that we saw in myogenic cell culture. We saw the protein go up, and then we used these drugs in mice, and we determined how effective they actually are at ameliorating the disease progression in mouse models for the muscular dystrophies. So in this case, we have a mouse model for Duchenne muscular dystrophy that we've used. We're seeing a large increase in the levels of alpha-7 integrin, and this is true in diaphragm, gastrocnemius, presumably a bunch of other muscles that we've looked at. We're also seeing a functional improvement where we are actually improving the strength of the muscle by increasing the levels of alpha-7. So now, not only are these mice going to be more healthy, but presumably in patients, they would actually live longer, because the stronger the muscles, the longer you can keep those muscles around, the longer these patients are going to live. And so we've also looked at how this is occurring. And one of the ways we think this is occurring is actually through enhancing muscle regeneration. These drugs that target alpha-7, at least the two that we've looked at so far, are targeting muscle regeneration in order to accomplish this increase in alpha-7. By targeting muscle regeneration and kicking that pathway back on, we're actually going to improve. We can presumably expand out into a lot of other areas other than muscular dystrophy as well. And this is the journal in which this work was actually published. All right, so here's the question we get quite a bit, is how are these drugs that are targeting the increase in alpha-7 integrin going to help patients with FSHD? Because I've transitioned into now using these compounds in an FSHD mouse model. And the key, again, is through myogenic regeneration. And so here's some work we've done in the FSHD mouse model. This is just an overview of the genetics of how the mouse model works. As I said, the DUX4 protein is extremely toxic, and so we've had to manipulate, using transgenic means, manipulate the mouse into not expressing the DUX4 protein until we want it to. And so we actually use a method where we use tamoxifen to flip the entire gene in the muscle cells in order to accomplish this goal of having it no expression to a lot of expression. So the bottom here is the treatment schedule for how these mice are treated. We began treating with our drugs, sunitinib or Striker 001 at 7 weeks. We then treated with tamoxifen at 8 weeks. And you can see this is the presumed amount of DUX4 expression in the muscle. It should be mainly gone by 11 weeks. Most of the cells that have expressed it should have died off, or at least the nuclei that are expressing it should have been dealt with. It's not completely true in the mouse because the mouse is able to deal with a little bit more toxic protein than we are as humans, but that's the idea. So when we did this, we saw a weight loss at 10 weeks of age in the tamoxifen-treated animals, which we predicted we would see because the mouse has lost all of that muscle. However, with our drug, in both males and females, we have seen an improvement in grip strength and in muscle force production in the blue. And so our drug appears to be working, it is regenerating the muscle, and so ideally we can actually get this into clinical trials, and it is actually an FDA-approved drug, so if we can raise the funds, we can actually get it into clinical trials relatively quickly. So here's a little picture. It's a nice pretty picture with pretty colors. It's a little washed out. We always like pretty colors as scientists. So this is what the muscle looks like with no tamoxifen. You can see nice long striated fibers. And this is what it looks like when it is on, when it's only treated with the vehicle, not the drug. You can see it's pretty beaten up. And then finally, you can see with our drug, we've improved it. It's not quite back to completely no DUX4 expression level, but it is definitely improved relative to the vehicle alone. And so that's my talk. I figured I'd field a bunch of questions about whatever you guys want to ask me, and hopefully that was a high enough level for everybody.
Thank you, Ryan. So Dr. Birkin's work actually coincides a lot with Ryan's work. So we are going to let Dean talk, and then we'll field questions about both talks after that, and then we'll transition to our other speaker who is switching gears kind of, so we don't want to confuse you. So Dr. Burkin is a professor in our pharmacology department. He is one of my favorite people to work with personally because his laugh is infectious. And I'm fortunate enough because I just got back from a conference today where I got to watch his wife, who is also a scientist, speak about her journey as a woman in science, which was really exciting. So I'm happy I get a day with the Birkin. So please help me welcome Dr. Dean Birkin.
And I met my wife, who's American, and so I'm here. There's the reason why. My talk today is about The Matrix Reloaded, and it's because I'm a fan of Keanu Reeves and the Matrix trilogy. I love Keanu. He's got some good moves. So that's why I've got this slide up. But basically, The Matrix Reloaded comes from the fact that, as Ryan mentioned, there are quite a few different muscular dystrophies, and many of them are caused by mutations in genes that encode proteins that bind the cell to the extracellular matrix. So the idea that we've done, the therapeutic approach that we do, is to try and restore that matrix. So that's the matrix reloaded part here. So we're developing novel therapies for the muscular dystrophies. So first I have to have a statement, disclosure statement, that we've got patents that have been issued for laminin 111 protein therapy, which I'll talk about today, that was licensed to a company in Boston, and that technology is about to enter clinical trials. We're within a year of clinical trials with that drug. And then Ryan talked about the small molecules that we've developed. Again, patents have been issued on those small molecules, worldwide patents, and we're moving them forward. Stricogen is one of the companies that has that. So Duchenne muscular dystrophy, this little boy has Duchenne muscular dystrophy, is a very severe muscular dystrophy. It's also known as Jerry Lewis's kids, Jerry's kids. And usually the kids are born normal. You can't really tell that there's any problem until they're about 3 to 5 years of age, and then they show developmental delay. So they have problems walking, they often have foot drop. In this case, this boy shows Gowra signs, where he has to use his hands to get himself all the way up. And they usually fall down a lot, and usually the parents take them to a pediatric, usually to their pediatrician, and then they take a blood test and they find out that they have very highly elevated levels of an enzyme called creatine kinase, which should be in your muscle cells, but it leaks out because the cells are damaged. And so then they're referred to a pediatric neurologist, and then they do a muscle biopsy and find that's really how you diagnose it. Unfortunately, the prognosis is really poor. The children are in a wheelchair by the time they're 12 years of age. They often require a respirator to breathe because your diaphragm is a muscle, and that is also degenerating. And then they usually die in their 20s or 30s from either cardiac failure, most of them about 90% from dilated cardiomyopathy, or from respiratory insufficiency, and then they get infections, which leads to their death. So unfortunately, you can't see this very well, but this is actually a section through the muscle here, and this is an unaffected person, and the dystrophin is in a brown stain, and it's found all the way just under the muscle fibers. So these are long fibers that are projecting out of the screen. And you can see that you have regular size. So all of our muscles have a fairly regular arrangement of these muscle fibers that bind together. So when you go and work out or walk, they're all operating together and contracting. But in this patient, what you see is there's like no organization, and there's holes and gaps in the muscle, and that progressively gets worse. As Ryan said, that they try to regenerate their muscle, but eventually regeneration fails. And so they end up actually losing that regenerative capacity. So it's the most common human X-linked neuromuscular disease, and it's X-linked, so therefore it affects boys. Girls have 2 X chromosomes, so you always have a spare tire. You have a spare good copy, and it compensates for it. It's a recessive disease. Us boys have that little Y chromosome, which doesn't really do too much. And so when we have a defective X gene, dystrophin gene on the X chromosome, we actually have it. So that's why it affects boys. Girls can have it. They can have a form of the disease that's a little bit delayed if they're a carrier. So DMD patients suffer from severe progressive muscle weakness. So as I mentioned, when they're born, you can't really tell. Everything looks normal, and progressively it gets worse and worse. And then they die in their second or third decade of life of cardiac or respiratory failure. There's no cure. There's no cure for this devastating neuromuscular disease. There's some really exciting therapies that are coming out. The current therapy standard of care is prednisone, and anyone who's been on prednisone knows the negative effects, especially if you're a child on prednisone, high-dose, long-term prednisone therapy. But there's some really cool therapies coming out. Exon skipping, which is a brand new drug coming out, and as Brian mentioned, gene therapy, and also CRISPR-Cas9 is coming on board as a gene editing mode for Duchenne muscular dystrophy. The DMD patients and the MDX mouse, the mouse model that we use, they have mutations in the dystrophin gene. These are naturally occurring mutations. In fact, you see a form of Duchenne muscular dystrophy through the entire animal kingdom. There's a dog model, a golden retriever, that naturally gets Duchenne muscular dystrophy. It's similar to that as well. So as Ryan mentioned, this is a similar thing, cartoon. There are major laminin binding complexes in muscle. The dystrophin complex is here, with dystrophin forming a scaffold. And when this is missing in these patients, when you have Duchenne muscular dystrophy, the entire complex is missing in these patients. So your membrane of your muscle cells are very fragile, and when this protein complex, which actually protect it, is missing, and you contract, when you generate force, you get holes punched into the cell, and then things leak in and leak out that shouldn't be leaking in and out. One of them is the creatine kinase that I mentioned. But when you get, if you get mutations in genes in any of these different protein complexes, you get some form of muscular dystrophy. As Ryan, limb-girdle muscular dystrophy is caused by mutations in genes encoding these sarcoglycans, and there's groups of patients that have sarcoglycanopathies. And I'll talk a little bit more about myrisin. Mutations in genes in the extracellular matrix cause myrisin-deficient congenital muscular dystrophy. And then alpha-7 integrin, we also have patients with those, and they're considered really ultra rare, but we've actually just identified a group of patients in Europe, and we've been working with a group of physicians in London, and we're actually characterizing those patients right now. So the hypothesis Ryan mentioned is that when this is missing, which is really like superglue, what happens to this other complex, which is just like Elmer's glue, and it naturally gets upregulated. So the idea here is that nature's already kind of showing us the way, and so let's go ahead and sort of follow what nature's showing, that if we can upregulate this even more, maybe we can rescue. So these are some of the papers, and Ryan mentioned them, to show the proof of concept. So we use transgenic mice, so I made transgenic mice in which I put a foreign gene into the mouse and expressed it at high levels in muscle. And when we did this, either the mouse was, without the transgene, was really sick and looked like a dystrophic patient, It had joint contractures, which are quite painful. And when it had the transgene overexpressed it here, you could see it restored the phenotype of the mice more like wild type. And then survival here just shows without the transgene, all the mice died at a young age, and with the transgene, they survived out about 3 times longer. The alternative is that if you lose the integrin, so you lose both complexes, you would predict what? So you've got the dystrophin and integrin, and if you lose both of them, what do you think would happen? Much more severe, right? You would expect that, and that's exactly what happens. So this mouse actually lacks both dystrophin and integrin, and you can see how small they are compared to just a dystrophin deficient mouse. And all these animals die at 4 weeks of age. None of them survive longer than 4 weeks. A mouse normally lives about 2 years, right? So they all die at 4 weeks of age. So this would really, these 2 pieces of evidence shows us that the integrin is a modified disease and therefore a target for drug-based therapy. And as Ryan already pointed out, we use this scheme where we isolated muscle cells from a mouse with a reporter gene, and we could screen drug libraries. And so the way that we did it was we actually collaborated with NCATS at NIH. NCATS stands for the National Advancement of Translational Sciences. It's an institute of NIH, and they have like a Boeing aircraft factory that 24 hours a day is screening drugs. That's all they do, screening libraries of drugs. And so they screened the drug libraries, and as Ryan mentioned, we screened over 430,000 compounds. Each compound was screened over 7 dilutions, so we get a dose response for each drug. So when we did that, we used laminin 111 as a control. And so laminin, when laminin binds to the integrin, it calls for more integrin to come to the surface, right? And so therefore it's a good control. And then we identified these compounds. And one of them was SU9516, which Ryan talked about. And SU9516 we published here is a failed leukemia drug. So it was already approved, so it went through clinical trials but didn't meet the endpoints for the leukemia. So we are currently trying to retool it for muscular dystrophy. There's a toxicity profile with it, so we're trying to resolve, so we're trying to change the molecule. But as Ryan mentioned, we increased the integrin substantially with this. And the good thing about it, it's very soluble and it can be put into a syrup. So when you think about children taking it, you know, if you've ever fed a child a tablet, they don't want to take it. Our kids didn't want to take a tablet, but they'll drink a syrup, right? And so hopefully we can put it in something that is reasonably tasty, that they can drink the syrup. So I'm going to turn to a different dystrophy. This is laminin alpha 2 congenital muscular dystrophy, and this little girl has laminin alpha 2. And so this is actually from a picture from NIH. It's caused by mutations in the laminin alpha 2 gene, and it results in the absence of that external laminin that's found on the outside of your muscle cells. It's characterized by severe muscle weakness from birth, so it's called hypotonia, and delayed motor milestones. There's a peripheral neuropathy, so laminin is critical for myelination of your motor neurons, and when that's missing, they don't myelinate properly, and so what happens is that eventually the nerve conduction goes down. So it's a dysmyelination. They also have changes in brain white matter. Although these children are super smart, we do the IQ test, they're higher than the average population. They're just super smart kids that are losing their ability to be able to walk. And most of them are in a wheelchair at a very young age, 5 or 6 years of age, and most of them die around 10 to 20 years of age. And they usually die from respiratory failure. There's no treatment or cure for laminin alpha 2 CMD. So there's nothing available, nothing at all out there. At all. Unlike DMD, where there are some things coming online, there's nothing available for these children. So in this case, we have our laminin on the outside of the cell in a normal, unaffected individual, and we have the integrin or dystrophin, and there's signals that go to the cell. When it's engaged in this way, it's actually sending survival signals to the cell and allows it to contract against the muscle. When the laminin is missing, the integrin is also missing off the cell surface. So you're missing both of those, and so therefore it's a double whammy, right? That you're getting double hit. So one of the hypotheses we want to test is that there's an embryonic form of laminin 211, which is missing here, called 111, which we could actually produce in very large amounts. And it's embryonic form. So when we're developing as an embryo, our muscles are surrounded by laminin 111, and it transitions to laminin 211, and that's the main form of laminin that's in muscle. And so the two are very similar. 111 and 211 are extremely similar in terms of their structure. So the good thing is we can produce a lot of this. It's really hard to produce a lot of this, and that's why we used it. So the experiment that I got my graduate student to do was, I was interested in regeneration, so I said, well, just inject some of the laminin into a muscle, and see if you can see regeneration around the edge, because it's a pretty big protein. And she came back to lab meeting and said, it went all through the muscle. And I said, go back and do it again. I don't believe you. But then she showed me this picture, right? So this is laminin alpha 2. This is 211. This is the adult form of laminin. And it's present in wild type, and it's missing in the mouse model because it's got the mutation. And then our laminin alpha 1, which is missing because it's obviously in the adult because it's an embryonic form of laminin. But when she injected it, she found it all around the muscle fibers. And so that was the shocking part of this experiment, because we wouldn't expect this protein, which is so big, to do this, but maybe not so shocking after all when you think about how this protein is made. It's made within the muscle cells. The muscle cell just dump the laminin into the extracellular matrix, and it self-assembles, and it looks like a cruciform. It's got a cross shape, actually. And so these cross-shaped molecules actually link to each other, and they form a gel, a matrix around the muscle cell. So this is actually the experiment where she took the laminin from a tube. You can see it go through the serum, so it's for systemic delivery, and then end up in the muscle. So you can see that you can actually follow the actual kinetics of the drug. So this mouse model actually replicates the human condition really well. It's a very severe disease. It said a mouse normally usually die between 8 and about 16 weeks of age, and they look miserable, right? They're very sickly, and they have, this is the peripheral neuropathy, and they don't groom themselves, so they look really mottled. This is a 10-week-old mouse now on laminin 111 protein therapy. We're just injecting the protein once a week, and you can see that his hind limbs are underneath, and he looks a lot better. And this is over a year. So this is the survival curve for the mice that didn't receive the drug. They just received vehicle. This is the survival curve of the animals that received the drug, right? So it's a transformational therapy, right? Because it's really increasing the life expectancy. And then if you actually look inside the muscle, remember these should be highly organized. This is without just with PBS, which is the vehicle. You can see all the damage in the muscle fibers. They've got big holes and gaps, and the fibers are a different size. And then this is with laminin 111 protein therapy. Not perfect, but it looks a lot more like this than that. So we can see we're preserving the muscle itself. And I think we've got a movie. He's going to play it, because I don't think it's going to play up here. So you can actually see them. So these mice are small. They're probably about half the size of a normal mouse. And so you can really see how really sick they are, which really replicates the human condition a lot. They have the peripheral neuropathy, dragging of the hind limbs. They tend to use their forelimbs are not quite as affected. And then if you actually look at that muscle, it's extremely damaged, okay? And it gets progressively worse. So we feed these animals, we provide water and food on the bottom. We usually group them together so that they can keep warm. So we just try to nurse them along, but they're very sick, just like the patients. The children are extremely sick too. And they usually have cycles of respiratory infections that eventually succumbs. They also have severe scoliosis. Your spinal curvature is maintained by muscle, and that also is affected in these patients. So often they require surgical interventions to straighten their spines. And then, so you can see, really can't even pull his leg underneath him. So this is a litter mate that's on laminin 111 protein therapy. Same age. Right, so you see the activity. So we haven't fixed the size, they're still small. There is a GI problem that we can't fix with laminin replacement, but you can see his activity. He's actually standing up on his hind limbs, he's grooming himself, his hair looks great, because they like to groom. So I think I need some laminin to stop losing it. But the grooming is probably the reason why they're looking really good. And their strength is also improved as well. So we look at their grip strength and so forth. Okay, so we can move back to the slideshow, get a pretty good idea. Okay, so to conclude, we've used a novel muscle cell-based assay to identify both biologics and small molecules that increase the integrin. The integrin small molecules have shown efficacy in the MDX mouse model, and we're hoping to move some of those to clinical trials. Laminin 111 protein therapy is already well on its way. They've managed to make recombinant human laminin 111 now, and they're at the clinical trials planning stage. And that'll be done most likely at NIH at this point in time to get the patients out there because they're so sick. And that these integrin therapeutics can be used alone or in combination with other therapeutics that have been developed. They can also be used for aging muscles, and we've shown with our transgenic studies that aging muscles benefit from increased integrin, so it's quite possible that there are other things. Also cachexia, which is wasting of muscle caused by cancer, that these could have impacts on. So more broad ranging than necessarily the rare diseases. And then finally, my team, I've got a lot of people who have graduated as graduate students and gone on to do postdocs. Some of them are at Harvard, others at NIH, and they've gone to all over the world. My current group, and then all my collaborators, I have strong collaborations at UCLA, at Harvard University, at Boston Children's Hospital, at Texas A&M, and then NIH. And then, of course, these are the grants that have supported the work, and this is our team here. So thanks a lot.
How cool are my coworkers, right? Like, the best. So we can open it up to questions for Dr. Burkin and Dr. Wuebbles at this time, if anybody has any. Yes.
That's a good exam question for medical students because you can set up a pedigree and say, okay, they had an uncle that died of a respiratory failure or a cardiac failure at a young age. So you can follow it through families as well.
When they do all the testing now for women that are pregnant, especially older women and all, is this one of the things that they discover?
I think Duchenne, they're beginning to do testing.
Duchenne is
just starting for congenital, no. I forgot to mention, Duchenne, the gene for it is the largest gene in our body. So it's the size of the E. coli genome. And so there's a target for mutations. So there are mutations across the entire dystrophin gene. And they can be point mutations, they can be deletions, they can be all kinds of different types of mutations. And it's really hard to standardize tests for testing it. That's one of the reasons. And laminin is also huge. It's a massive gene. These are large structural proteins. And so they're also difficult to be able to identify and test with any rigor.
So there's a 1,000-fold difference in the prevalence to, 500-fold difference in prevalence because Duchenne's is 1 in 5,000 boys, and limit to CMD is actually about 1 in 500,000 people, period, to 1 in a million. It's extremely rare. For FSHD, it's actually a dominant disease, so it's 50-50 shot of a parent passing it on to their children. I actually did what's called PGD, pre-implantation genetic diagnosis, in order to make sure that my 2 boys did not have it. And so it's a way to actually screen out and make sure that you don't pass certain things on. But it's the contraction, as I said before, it's highly variable. My brother actually has the same exact mutation or contraction in the repeats that I do, and he doesn't have any symptoms at all.
Yes.
If you're doing gene modification, is it permanent or do you just have to keep doing it?
So that's a very good question.
Good ethical question.
Yeah. Currently, because the AAV delivery is presumed to maintain these as extra chromosomal, they should not integrate, and it's believed that eventually you'll need to replace them. It's not clear how often, because the clinical trials have just gotten underway. And in dogs, I believe they last, the average lifespan for the expression was around 2 years. So 2 to 3 years. So if we presume that our muscle turns over a little more slowly than dogs do, we would predict probably 4 to 5 years, but that's just the predictions I've heard.
Yeah, so the only way to do it is to deliver it with a virus, right? So if you're doing CRISPR-Cas9 gene editing, you have to use a virus. You have to use 2 viruses, in fact, to do that gene editing. And for the dystrophin, where they're using a sort of a shrunken version of the normal dystrophin gene to package into the virus, that is one shot with the virus. But you can imagine, viruses, you get immune response to a virus, right? And so there's the problem with the immune response, both for CRISPR gene editing as well as for gene therapy, and that's a major issue that has to be overcome.
Yeah, that's actually the larger issues. We only have a certain number of viruses or AAV that will actually target muscle. There's only basically 3 that I think do it fairly efficiently. And so you can imagine if we get a complement activation, which is a massive immune response that's life-threatening, and we've already seen that even in children with the current microdystrophin that's been delivered by Pfizer and by Solid. They've had about 50% of their patients that have received AAV9 actually had a massive immune response. And adults are going to be much higher percentage than that to AAV9 because we've seen a lot more
viruses in our lifetime.
Yeah.
We've been infected more times.
So yeah, I would say that it's a bigger problem to find or engineer new viruses that will actually target muscle because we don't know whether we're going to be able to pump a patient full of the same virus. AAV again.
It obviously takes a long time from doing research in the lab and coming up with a promising solution to clinical trials and actually out in the world. So how long does it typically take?
That's a really good question too. It can depend. From the business side of thing, having talked to a lot of pharmaceutical companies, it sounds like 15 years is about the average. We know of drugs that have taken. We know of drugs that we saw results in a paper from 20 years ago, and they're still not even close to clinical trials. So it can obviously take a lot longer. And there's some things that people can just rush through as well. When you already have a drug that you're retooling for that's been used for a different purpose, such as the sunitinib that we showed, it's already FDA approved, so you can actually get it through the FDA process much more quickly. There's a lot less hoops to jump through because it's already been proven to be safe. You just have to prove that it's effective, and even that, the FDA doesn't care as much about it being effective as it does that it's safe.
Are you going to change your battery? Okay. So I'm going to give Ken an opportunity to change the battery on his camera. And we're going to switch gears a little bit. But to add to the answer for about how long it takes things to get to clinical trials and to market, a lot of that is dependent on the need and how big the need is for any given treatment. So I talk a lot with Dean and Peter Jones, who's the FSHD researcher at our institution, and it depends a lot on where the money is to develop it from being in mice to getting it to people and then running through the stages to make sure that it can get to market okay. The safety stages and the efficacy stages and the dosing stages, it takes a while to run all that through, and it costs a lot of money to do that. So that's also a barrier.
Danielle?
Yes.
One thing too, Dr. Connor's going to be talking. I'm going to pass out some of our cards that we normally have, for questions. And I'm going to ask you, this is kind of a little survey. We're trying to update our marketing and all that. So we're asking for testimonials. So I'm asking you to write just maybe a 1 or 2 sentence testimonial on this forum. And also, the second question would be, what other topics would you like to hear about from the UNR research area? What is interest to you? So 2 things, and I'll pass it out while he's discussing, okay?
Yeah, and I will have business cards after this because I would love to hear from you. Just if you have questions after this or want to know what's going on at UNR Med, if you'd like to come up and get a little baby tour, I would love to meet you downstairs at the lobby of my building and give you a little baby tour to show you kind of what we do up there. Because it's really, like I said, it's really exciting. Can you tell I love my job? It's like the best ever. Are we good? Okay. So we're going to shift gears a little bit. Dr. Kenneth Hunter is a professor in our Microbiology and Immunology department, and he's an immunologist. And he is, a little personal note, he was one of my undergraduate professors, and he was one of the people that taught me kind of how to love science and how to explain science in a way that is reachable for people who might not love it as much as I do. So, Dr. Hunter, you're on.
So I'm going to talk to you about cancer immunotherapy. I'm going to show very little of our own work. This is more of a general introduction to something that is really exciting and new. And I hope it's relevant to all of you, and if you don't think it is, in a matter of seconds, I'm going to make sure that you think it is. Okay. 39.3% lifetime risk of getting cancer. Let me put that in another way. 1 of every 3 of us, more than 1 of every 3 of us in this room, will have cancer in our lifetimes. Isn't that amazing? Now, a lot of you are a little bit older, like me. I haven't had a serious cancer, but I have had some skin cancers, so I already fit in the having had cancer business. So it's really amazing. It's the second leading cause of death in the United States, right directly behind heart disease. Yeah, right behind heart disease. No. Let me just show you a couple other statistics here and we'll move on. Men have a little bit of a higher risk of getting cancer, and it's because we have occupations where we're subjected to carcinogens more often. More often we're outside where the sun basically hits us. Men actually smoke more. All of those risk factors, but women are right there. It's not such a huge difference. If you get cancer, even today, the chance of survival is not great. There are cancers, so you just take a look at this list. Just let your eyes go over it. If you get non-small cell lung cancer, you've got about an 80% chance of dying. If you get skin cancer, you're probably not going to die. But all in all, if you look at all of the cancers, across all of the cancers, if you're diagnosed, there's a 72% chance you won't live more than 5 years. So this is a very, very serious disease. And we're going to talk today about something that is really new and really exciting that could have an impact on many of us in this room. Okay. Well, we're not going to go into great detail about what is cancer. I mean, obviously, most of you understand this. It's the unfortunate consequence of what happens when perfectly normal cells in your body that shouldn't be growing uncontrolled start growing with no control. That's the notion behind cancer. It is, there's a lot of different causes for cancer, as I mentioned, many risk factors for it. But in the end, what you're talking about is a situation where you have cells in your body that shouldn't be growing rapidly, and they do. And sometimes it just forms a large benign lump, and it's not serious. Other times, well, if that large benign lump is in the middle of your brain, that can kill you. But oftentimes, cancer will actually be metastatic, it will be malignant, and will spread throughout the body. Now, some of us acquire genes from our parents that give us an increased risk, but generally speaking, it's our environment that is the greatest risk factor for cancer, so keep that in mind. Okay, cancer treatment. Even today, as it was 30, 40, even 50 years ago, chemotherapy, radiation therapy, and surgery are still the mainstays of cancer treatment. Still the mainstays. And what's important for you to know, living in Nevada, and many of you probably have already experienced this, if not directly, In your own personal life, you know of circumstances where people develop cancer, and they ended up having to go over to California to get their treatment. And some of that still happens now, but we have developed in Northern Nevada, through the School of Medicine in cooperation with Renown in Reno, we have developed some extraordinary capabilities in oncology. So now, if you need very sophisticated radiation therapy, you can get it right in Reno. If you need to get the latest in chemotherapeutic drugs, you can get the treatment right in Reno. And the latest in surgery for cancer right in Reno. But that's not what I'm going to talk to you about today. There are great advances that are occurring in those areas as well. But I'm an immunologist, and what I'm going to talk to you about today is cancer immunology and the development of cancer immunotherapy. This is very important and it's very exciting. And I'm almost sad to say that many of us in this room will become familiar with this. And so let this, if you haven't heard about it, be your sort of first exposure to it so you have a sense of what we're talking about. The immune system. When you think about the immune system, you think about microorganisms. Still, on planet Earth, infectious diseases kill more people than everything else. Of all the people who've ever lived on our planet and died, of all the people who are living now and will die, and for all the little babies who will be born, live, and die for the next 100 years, I predict infectious diseases will still be the most important cause of death. But let me tell you this. If you didn't have an immune system, you wouldn't be sitting here listening to this talk. If you don't have an immune system, you can't live in with the microbial world around you. That's how important the immune system is. And for years and years and years, we thought that maybe the immune system might have something to do with cancer, but we weren't sure. So in 1960, Nobel Prize was given to Sir Macfarlane Burnet, and he had hypothesized that the immune system should be able to interact with cancers that develop in our body. Do you see that sentence there? Yeah. Harmful non-self and harmless self. What on earth does that mean? Well, your immune system evolved to protect against harmful things that aren't you and to ignore you. Do you think the immune system is always effective and never attacks self? Well, of course not. You know the autoimmune diseases, sometimes the immune system makes a mistake. But actually, even those are very bad diseases, it's pretty rare. So think about the problem with cancer. If the immune system actually is going to help us get rid of our cancers, cancer derives from self. If you have a lung cancer, there was a cell in your lung that received a sufficient number of mutations that it could begin to proliferate in an uncontrolled manner. It's now a cancer, but it derives from you. Your immune system is supposed to be ignoring self. So the only way that your immune system can help with cancer is if the cancer cells begin to appear as non-self. And thank goodness that's the case. Years ago, when I started doing research in cancer, I used to look at tumor cells taken from patients, and sometimes they were big and gnarly and ugly, and you would just say, oh my goodness, this is just terrible. And oftentimes, those cancers were eliminated by the immune system. You know what the worst cancer is? It's the cancer, the only thing that changes in that cancer is that it loses growth control. Everything else stays exactly the same. And what does your immune system do? It just ignores it. Those are the cancers that kill you rapidly. But we now know, thanks to a lot of work in thousands of labs for decades now, that the immune system plays an important role. As a matter of fact, we use the term immune surveillance or cancer surveillance. Your immune system recognizes tumors, oftentimes when they first appear, and eliminates them. For all you know, in the last 2 or 3 days, each one of you developed a cancer in your body that was immediately recognized by the immune system, and the immune system is in the process of knocking it out. So we now know through experiments that that happens. Okay. So for most of us, when I say the cardiovascular system, what do you think? Do you think about the heart and blood vessels? Well, the immune system actually has an architecture. Lots of immune cells are found in our lymph nodes, for instance. The spleen is another good example of an immune system organ. And actually, almost all of our immune system originates in our bone marrow. Our bone marrow makes most of the immune system that keeps us alive in the microbial world and does battle with our cancer cells. So, because time is short, and because, as Danielle knows, having taken my full semester immunology course, which is 44 hours of dreadful lectures, we have 20 minutes, and so we're not going to go into all of this. But for cancer immunology, I want you to think about 2 things. Antibodies. You know about antibodies. Your bodies make antibodies to protect you against influenza virus and group A strep throat. But you probably don't know as much about T cells. They belong to a family of cells in your immune system called lymphocytes. And we're going to be talking just in a moment about antibodies and T cells. And I'll explain that just a bit more. The immune system is very complex. You saw some of the research work that is being done on muscular dystrophy, and I'm sure some of that went over your heads. Well, in almost every bit of research that we do in the School of Medicine, it's that level of complexity. So the immune system is very complex. As I said, we're only going to be talking about the cells that make antibodies and the antibodies themselves, and lymphocytes, and in particular, you're going to hear me use the term cytotoxic T cells. Okay. Upper right, that's a picture of an antibody molecule, and that's a cytotoxic T cell. So let's see what we can do with those. Research all across the world for the last couple of decades has led to some amazing discoveries that are saving an enormous number of lives in people who have cancer. As I said before, if you get cancer, oftentimes you will have chemotherapy, you will have perhaps some radiation therapy, maybe if your tumor is reachable, somebody will go in and remove it. But even with that, the carnage caused by cancer is still huge. So we need something else. And so scientists working at our medical school here in northern Nevada and all across the country have been working to make the immune system more aware of cancer so that our immune system can actually be used to eliminate cancers that show up in us, and in some cases, perhaps even prevent cancers from showing up in the first place. We're going to talk about monoclonal. This is a special genetically engineered group of antibodies that are being used regularly now all across medicine, but most certainly in cancer. I'm going to tell you about something called adoptive T cell therapy. This is something we do in my lab in collaboration with oncologists all around the country. And some rather spectacular results have occurred. Cancer is simply being eliminated. We'll talk about that just briefly. Immune checkpoint inhibitors. What does that mean? Well, I'm going to show you it means a lot, and it meant a lot to the Nobel Committee, who gave the 2018 Nobel Prize to a couple of people who invented this. That's right. We can now make cancer vaccines. You can be vaccinated against your cancer. And at the end of this talk, I'll talk about something I'm doing in my lab, which is really an interesting area of endeavor called oncolytic viruses. Therapeutic monoclonal antibodies. Monoclonal antibodies, antibodies that let me see if I can explain this in something in one way that's not technical. I can reach into you, and I can take cells from your body that make antibodies, and I can immortalize them, and I can isolate the very specific antibody that I want, and I can make that in unlimited quantities. Unlimited quantities. It's called monoclonal antibody technology. And it's more than 35 years old. I know. Because I made the first one. Now, there are over 500 clinically available human monoclonal antibodies, many of which are used in cancer. It's really quite the rave. The example that I'll use is cetuximab. If you have a head and neck cancer, or if you have lung cancer, if it happens to have the epidermal growth factor receptor, EGFR, which is shown over here, you can take a drug, which is a monoclonal antibody, which binds to that molecule on the surface of your tumor cells, and it prevents those tumor cells from receiving a growth signal, and they stop growing. Very widely used. I wouldn't be the least bit surprised that somebody in here has actually had cetuximab for a cancer, because it is a widely used drug, and we use it in northern Nevada. What other uses do we have for monoclonal antibodies in cancer? It's much more extensive than this, but just imagine if we could make an antibody, we could identify an antibody. Remember, I can reach into you and pull out that antibody and make it in unlimited quantities. And of course, the one that I want is the one that binds to the surface of a novel molecule that has shown up on the surface of your cancer cells. And that antibody can tag your cancer cells so that your immune system can recognize that as something that shouldn't be in the body and can remove it. And that's basically shown here. I can take an antibody to a cancer, and I can put an anti-cancer drug on that antibody. I can actually chemically conjugate that. And when I inject the antibody, it finds your tumor and delivers the anti-cancer drug to your tumor. What do you think the biggest problem with most cancer chemotherapy is? Side effects. Most of the drugs we use for cancer chemotherapy simply kill dividing cells. Well, there are perfectly fine dividing cells in your body that shouldn't be killed. But when you take that anti-cancer drug, even though it may kill your cancer, it'll also knock out your dividing cells. Why do you think the hair falls out in that patient who is having chemotherapy? Why do you think they have gastrointestinal problems? Well, that's where cells are continuously growing in the human body. We can take that antibody and we can put a radioisotope on it. A radioactive isotope. When we inject that in, it finds the tumor, and then it irradiates the tumor. You don't have to have radiation from a large machine coming into you. You can actually deliver the radiation directly to your tumor. All of this is made possible by human monoclonal antibodies. Now here's a monoclonal antibody. It's used in cancer. It's not very good, actually. But I do want you to notice something about this. If you were to take this for your cancer for a year, it would cost you $178,000. You know, this big controversy about the cost of prescription drugs and so forth. But you also heard that it may take up to 15 years to develop a new drug and get it through the FDA. I worked on a monoclonal antibody in the early days that took 8 years and $350 million to develop. Now, when that antibody came out, and it had a price of $3,575 per dose. I thought, this is ridiculous. But if you did the math, that would still take, the company that developed that antibody would still take them the better part of 30 years to recoup their investment. Did you see what I'm saying? Medical research is very expensive. Adoptive T-cell therapy. This is personalized medicine. And what do we mean by that? Well, in adoptive T-cell therapy, let's say you have a cancer. And you come to me and you say, oh, I've got bad news, I've been diagnosed with lung cancer. I say, well, unfortunately, in the modern era, I probably would next say, how much money do you have? Because it's very expensive to do what I'm about to tell you. But eventually it won't be. I can reach into you and I can isolate from you those cytotoxic T cells that could kill your cancer. And I can remove them, grow them outside of your body into billions and billions of cancer-killing cells, and then put them back into you. Could I see the movie? Are you up there? I can't see him. Cytotoxic T cells are the cells in your body that can recognize tumor cells and can kill them. And I'm going to prove that to you in this movie.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target.
The blue cell's the cancer cell.
Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate.
Boom, that's a dead cell.
Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
Okay, that's sufficient. Thank you. So let me repeat what I can do. If you have cancer, I can reach into your body. I can identify those cytotoxic T cells that kill cancer. I can take them out of your body in my laboratory, and I can expand them into billions and billions of cytotoxic cells. I can then put them back into you. They will find your tumor, and they will turn it red. Not really, but they will kill those cells. So take a look at this. It's kind of hard for me to turn like this. All right. So this is a patient who has breast cancer, or had breast cancer. And this is a CT scan, familiar with those? And this is a nodule of that breast cancer, and it's in the chest wall. And if you look at the liver, that's metastasis. The breast cancer cells left the breast tissue, went to the liver, and are setting up housekeeping as these tumors. This patient, however, received adoptive T-cell therapy. At 14 months, nothing in the liver and the breast tumors are gone. That's a complete cure. You do realize, of course, that in many of the serious cancers, if you have chemotherapy and radiation or even surgery, all it really does is buy you a few more months. This is a complete cure. Since we're a little short on time, let me just say that these immune checkpoint inhibitors that I had mentioned before, that's the term that they're given. There's an immunology professor that Danielle had who basically said it's as important to turn off immune responses as it is to turn them on. Immune responses are great. If you have a virus infection, if you have cancer, you want to have an immune response. But the immune system is so potent that after the immune system does its thing, you have to turn it off. And so we have evolved in our immune systems lots of ways to turn off the immune system. And that's a perfectly fine thing. Well, who was to know that some of the molecules that our immune system uses to turn itself off with, a cancer can begin to express on its surface. So now, tumor cells can turn off those cytotoxic T cells that are supposed to be killing them. Is that nefarious or what? So these two guys, Jim Allison and Chizuru Hanjo, they got the Nobel Prize last year, 2018, because they identified these checkpoint inhibitors and how they're working on tumor cells, and they came up with, monoclonal antibodies that can block the tumor from turning off the T cells that want to kill it. And so if you have a cancer in northern Nevada, it is highly likely, if it's a head and neck cancer or a lung cancer, you will receive one of these drugs which will prevent your tumor from turning off those T cells, so those T cells can then kill your tumors. This research started in the 1960s, and it's just now come to market. So how many years is that? That's a lot of time. We can make a vaccine. So if you have prostate cancer, we can actually give you a vaccine, because all prostate cancers begin to express this prostate acid phosphatase molecule, and we can make a genetic construct of that molecule, convert it into a vaccine, remove cells from your body, put those that vaccine into the cells and put those cells back into you, and those cells will turn on cytotoxic T cells that will kill your prostate cancer. It's an actual anti-cancer vaccine. One of the most important public health problems on Earth today is antibiotic resistance. All across the world, the microorganisms are becoming resistant to the antibiotics that we need to stay alive. But you know something? Tumors can do the same thing. And so the big problem for this cancer immunotherapy is that it selects for tumors that can avoid the immunotherapy. And that's what I'm showing you on this particular slide. Here are some immune cells that are recognizing the tumor. But then this tumor here has mutated, so the immune cells can no longer recognize it. And eventually it begins to grow into a mass of tumor cells. So even though cancer immunotherapy is fantastic, we still have a ways to go to figure out how we can get around the notion that as we're trying to develop ways of killing the tumor is trying to figure out ways of avoiding those killing mechanisms. Okay. So we just have a couple more slides here. We're going to end on something that we're doing in my lab right now that is really exciting for cancer. The research that's going on now is trying to figure out how we can basically get around the idea that cancer cells treated with drugs or radiation or these immunotherapies and begin to develop resistance to them. How do we deal with that? And there are lots of research projects that are showing some promise in that regard. combining therapy. So now it isn't, we didn't have immunotherapy to combine with other therapies until most recently, but now that we do have immunotherapy, we can combine it with radiation therapy. We can combine it with chemotherapy. And there are literally hundreds, perhaps even thousands of clinical trials going on in oncology right now looking at these combinations, and the results are looking very favorable at the changes that occur in all of the different cancers. You know, at the very beginning of this, I really didn't point out a feature about cancer that is a misconception in the broad public. Cancer isn't one disease. You know that. There's over 100 cancers, and each one of those cancers is individualistic. I can have lung cancer, you can have lung cancer, and they'll behave differently. Now, they behave more similarly than lung cancer and prostate cancer, but this is the problem. So many different diseases. And so we need to be able to get our arms around that. And of course, most of cancer therapy has side effects, and we're interested in eliminating that. If somebody was to ask me what one of the greatest things that's happened in cancer in a long time, and I've been around for decades, where I've seen cancer patients who just would look up at me and basically say, the treatment is killing me. I just want to die. Just let me die, okay? I've been throwing up for the last month. I can't stand it. And now, they're not anti-cancer drugs, they're anti-nausea drugs. And they basically make having chemotherapy and radiation therapy a much less problematic experience. I think, to conclude, that the hottest area of cancer research, this is my own opinion, is oncolytic viral therapy. Now, what did he just say? So many years ago, of course, there's nobody in here except for a couple of us who would have ever seen a case of smallpox since it was eradicated in 1976. But when the smallpox vaccine, yeah, some of you have had the smallpox. When the smallpox vaccine was given, one of the things that people noticed is that after receiving the smallpox vaccine, some people who were vaccinated who had cancer, the tumors disappeared. Are you kidding? Well, here's what we know now. There are many viruses, many different viruses, that selectively kill cancer cells and do not kill normal cells. Right now, there are over 60 clinical trials underway with oncolytic viruses, viruses that will kill cancer cells and not kill cell cells. The first one has been approved in the United States. This is Imlygic. It's actually for melanoma, and you actually inject the virus directly into the solid tumor, and the virus then kills the tumor and it doesn't kill the normal cells. That's amazing. What are the side effects? No side effects. Your hair doesn't fall out. You have to make sure that whatever the virus is, it can't grow in the body and cause viral problems. And of course, they've been engineered, like the one we're working with in my lab, which does the following things. I just injected that virus into your tumor. It's killing your tumor. And when your tumor's gone, since it can't grow in normal cells, all that's left is you.
So we'll take questions.
Does Medicare cover that?
Imlygic, the only one approved. By the way, the first oncolytic virus was approved and has been used for 30 years in China. We're not always the first. Imligic is covered under most insurances, yes.
Yes.
So since you raised the issue of costs, how are all of these, I mean, if you're not wealthy, how do these filter down to the average Mary and Joe Blow and taking it for whatever? And I think the medical establishment has to be one of the sources of information about this, not just the public.
Cancer kills more poor people than rich people. Do you know that?
I do.
It's just a fact of life. In Sweden, where they have socialized medicine for what it's worth, it doesn't make any difference how much money you make. The rate of cancer death is going to be the same. If you can't afford, a standard person who has lung cancer, going to the hospital, CT scans, surgery, radiation therapy, perhaps some of these new therapies keeps that person alive. Normally, in the old days, you'd be dead in 4 months, but now it's a year. It's a year and $2 million of medical bills. Yeah. But on the so we scientists, quite frankly, what we do is we go into our laboratories and we try to develop what's necessary to save lives. It's politicians and the public who've got to figure out how these modalities that we come up with, that are extremely expensive, are going to make their way to the people who need them.
And I agree with you, but those two entities have failed for the last 30, 40 years to do what you're talking about. Maybe I'm wrong, and maybe because I'm the public, I feel that you have more inside information about how costs can be changed or dealt with differently, say in Sweden or wherever, than we're doing it, because we're doing it really poorly. If money is the line between life and death, that's pretty poor.
Well, you know, it all goes back to money. I don't know, I can ask Ryan and Dean, I mean, how much grant money have you had over the last few years to do your work?
Maybe $700,000.
Yeah, I was going to say almost $350 million.
Yeah, that'd be my guess, which sometimes
Well-
makes us support 5.
Oh, I think it's more than that.
No,
Dean,
it's more
how much
than-
have you had?
So it's probably about, so I've had probably about $12 million over the course that I've been here, that I've brought in grant
How many
funding.
years is that?
It's over 16 years.
Yeah,
Yeah.
so $12 million over
Yeah,
16
that's almost the exact same amount that I've raised to support my work. Most of that coming from the federal government. And I can't tell you how panicked I get when they start talking about where they're going to cut the budget, and they start talking about cutting the NIH budget. And I would just say to the senators, you go ahead and cut the NIH budget. But when you develop prostate cancer, Senator McConnell, do the right thing and not take any of the drugs that we've developed, because without that support, we can't do what we need to do. But even with that, we develop the drugs, whether it's for muscular dystrophy or it's for cancer, and the pharmaceutical companies play a huge role. And you guys are just now getting involved. That virus, the last thing I showed you, that goes into clinical trials in November of this year, so it's just about ready to go. That's going to be years, and I mean, it could be $100 million, $200 million to get that approved.
The average cost to get a drug through clinical trials is in the $300 million range for especially for muscular dystrophy, for orphan drugs, where you're talking about a rare disease in patients with mobility problems and transportation and everything else. It can vary, but usually the more prevalent a disease is, the more rigorous the testing needs to be through the FDA, and so you're requiring a higher number of patients, a higher amount of a clinician's time in order to get there. And so, yeah.
Do you know the term that they use for some of the diseases like these variants on muscular dystrophy? They call them orphan diseases. Orphan diseases. Because the drug company looks at that. I mean, if I go to a drug company with an oncolytic virus that can kill, 15 different kinds of cancer, they're just sitting there, quite frankly, thinking, okay, this is going to be worth the investment. Okay, I'm going to be able to get my money back on that investment. Whereas in the orphan diseases, it's hard to know.
It's philanthropy driven.
So just in the most common muscular dystrophy, how many patients in the United States?
You're about 30,000. So
30,000.
about 30,000. And for the
Patients,
for
it's
Duchenne,
actually a little,
yeah, and
it's actually less than that.
and for congenital, there's only about 1,000 patients, which I talked about. So for those ones, like for example, SMA, which has the most expensive drug, $2 million for treatment for a gene therapy. It's the most expensive one. There's spinal muscular atrophy. That one is ultra rare too. So the drug companies have to charge that amount to recoup the money they've spent out. So.
Right now in the United States, there are 20 million people with cancer. So drug companies look at that as a market, you see.
There's one behind you, and I think we're running low on time.
Yeah.
Judy's going to pull us off. Go ahead. Yeah.
I spent time in Scotland and was absolutely amazed at the difference. They have medications that we've been testing for years, and they're using them with fine results. And their whole attitude is to make people well and give them as good a life as possible. And so they don't worry about how much it costs or anything else. If a person needs something, they will get it.
And who pays for it?
It comes from their, I've forgotten the name of the tax, but what most people pay for their taxes, for their medical, is probably a tenth of what we pay to our insurance companies.
Yeah, I think that, there's positives and negatives. So one example where the US is really superior is thalidomide. Remember thalidomide babies? So it was rapidly approved in the UK and New Zealand and Australia testing. And then that ended up to be a teratogenic compound that caused all these deformities in babies. In the US, it never caused any problems because the US FDA was very rigorous on the testing. They didn't accept that the companies hadn't done the testing. And so it was never really approved. And so there are very, very few cases of thalidomide babies in the US. So this is where FDA really took off as being sort of the premier of safety. And that's where the major thing is. So that's really where things have changed a lot, and why, for example, insulin was approved and used worldwide within 2 years of the basic research, and they've received it, where now it takes 15 years to get approval. The main thing is safety, not only efficacy, but safety.
Right.
Well, I get some of my medication from through the UK or from different parts of the UK, and I know there was one medication that they developed and I took, and it was fantastic.
Sure.
It was better than anything that was offered. It's just the whole attitude, though, was so different. This hasn't really been tested as much as we'd like. If you want to try it,
Yeah, I mean, there are definitely,
You know.
there are definitely circumstances where the FDA will rush something through or
The HIV
fast track
drugs is a classic example.
to fast track them.
Everybody, the outcry was just huge. This is an epidemic, we need to do something immediately.
But
Is there a question up here?
So can you talk to us about, did I hear there was some new law they were trying to push through that you could sign and say, go ahead and let me in on a trial, even though, let me try it to see if it'll save my life? How is that working?
Well, in cancer, there are clinical trials in cancer are one of the major directions that patients need to go in after they've gone through the first rounds of the common things. You can get the best of care, you are trying new things, but the FDA has basically said, once we have some preliminary safety data going forward in these clinical trials, monitoring the patients extremely regularly, we're going to let you move forward into clinical trials very, very rapidly. In the old days, the FDA basically would have drug companies go through 3, 4, 5 rounds of clinical trials before they would approve. And now, if there's some suggestion that it might be good, they'd like to have a little bit more work, they are saying, in some instances, all right, we're going to give you a transitional approval, and you're going to go ahead and use this drug, but you're going to monitor those patients carefully, and if there's any adverse effects, we're going to know about it, and we reserve the right to pull it off the market. Now, patients who take those drugs do have to sign and say they understand what they're getting into.
And related to that, if there is an investigational product and it seems like, and these are more prevalent in like huge cancer centers, if it looks like an investigational product that is being studied at that particular center, looks like it's going to show promising results for somebody who maybe didn't meet inclusion and exclusion criteria to be on the trial, but they want to try to use it as a life-saving, as a potential life-saving measure, there are mechanisms to do that through different consent processes and hospital approval. But that is, again, it'll be heavily monitored by the FDA for the clinical trial from a clinical trial standpoint from the person who's sponsoring that trial. There'll need to be, there's tons of documentation that has to happen, but there are instances in larger cancer centers where they can do that. And there's also a consideration of capacity there. So if you are feasibly on your deathbed, how are you going to be able to acknowledge and be able to legally sign and say, yes, please give me that investigational product, it's my last hope. So there's a set of capacity issues there as well in regards to doing that.
To follow up on her question, as far as, you said that they have to go through normal, like chemotherapy, whatever, surgery, whatever, first?
Not all the time.
Okay, that's what I'm getting at.
Yeah, not all the time.
Can you
So
go right into the viral treatments and all that?
it depends. It depends on the cancer.
It does, but the short answer to you is that under certain circumstances, you could go directly to the novel therapy.
Mm-hmm.
Okay, but oftentimes what they'll do is they'll leave the therapy, the standard approach will be done, and this is a bit unfortunate. I did a lot of research in my early days on a condition called septic shock, sepsis. It's what happens when people with infections, the infection becomes systemic. And it has a 70% fatality rate. And when the compounds that we were developing for that, we couldn't give until every other standard thing had been given, which meant we were in the last hour.
Right.
Okay, you get the last hour. So whatever you're doing, it has to be able to take somebody who's extremely debilitated. Well, the FDA has kind of backed off on that. They're basically saying, no, we need to get these, in their proper sequence. So you're not trying something in a person who is so far gone that the odds are it won't do anything.
I think someone down there has a question. Down in front? Nancy? Did you guys have
It was answered.
Anybody else? Any other questions?
Oh.
Has the adoptive therapies been used in any of the bone marrow
It has. It has. You're talking about leukemias and lymphomas. Yes, it has. Yeah.
Where they're giving infusions every so many months to build up the cells again.
Right.
Okay, anybody else? For any of the researchers? Okay, thank you so much.
Yeah.